Thank you guys so much for letting me speak. I'm sorry about the little technical difficulties and sorry that I'm not physically there. I'm actually in St. Louis, which I think is really apt because I will be talking about the stuff that and the suite of tools that I developed while I was a grad student in the Pincus Lab at Washington University in St. Louis. And so with that, the Pincus Lab, so in the Pincus Lab, we were interested in studying the aging process in C. elegans. And specifically, we're interested in sort of answering the question of nature versus nurture and sort of individuality. And like all living things, C. elegans change morphologically over time. And we were interested in quantifying those changes and understanding how differences in these changes between individuals can affect overall lifespan. So here, we have the same individual imaged from adulthood, from the first day of adulthood until death. And the worms here have been computationally straightened with the head over here and the tail on the right side, so that you can clearly see what's happening over time. And, oh shoot, dang it, I don't know what's happening. Okay, we are in the Pincus Lab, we were really interested in sort of quantifying these changes and at an individual level. So to do that, the Pincus Lab created this system, this single animal culturing system that we like to call the worm corral. And this allows us to capture images of hundreds of individuals in parallel throughout their lifetimes. So worms are housed as embryos in these little food pads, and then we image them on a microscope every couple hours from hatch until death. And I like to think of it kind of as like the Truman Show, but in parallel for worms. So just giving an idea of some of the data that we're working with here. So here are 20 individuals. They're genetically identical, cultured in as near an identical environment as we can. And these colored bars in the bottom here indicate different developmental processes and larval stages that they're going through. And you can see that even though these worms are as identical as we can get them, they still experience life in a very different manner. And so we're trying to figure out on the broad level, like, what is it that makes one worm different from another? And what this really breaks down into is really, how do we compare two worm images? So we have a lot of data to deal with the worm corral, and these worms can be in a variety of positions. So we're trying to identify, how do we compare this worm in this image here to this worm here? Now let's think of an example where, let's say you wanted to look at sort of the pharynx of these two different worms. So one way that you can compare two worm images is by manually identifying regions of interest. So we can draw a circle around the pharynx in the worm here and a circle in the pharynx of the worm here, and look at the pixels between these two worms. But, manually identifying points is really tedious. And so the main question and goal that I was trying to develop during my time as a graduate student was, we need to develop a method to establish point correspondence between every single image. And what I ended up doing was creating this pipeline. It's a fully automated pipeline that allows us to make these point correspondences. So first, we have to find the worm in the image. We have to straighten the worm to account for any positional variation. And then we align the worms to a standard frame of reference to account for any internal individual variation. And then from there, we can do a variety of different downstream analyses. And so how this works is, the first two steps, we take what we call the lab frame of reference, or the laboratory frame of reference, or lab frame image, which is the full image that we get from the microscope. We have to identify where the worm is in the image, indicated by this pink binary mask here, because it's easy for us as humans to know where a worm is, and it's much more difficult for a computer to. From there, we identify the center line and the widths of the worm, and the different axes that they're on, and then once we get the center line and the width, this is a pretty quick and affine transformation to go from a curved worm here to a straightened worm here, which we call the worm frame of reference, or worm frame image. And now we have put all of the worms into the same frame of reference, where we know all the pixels within a worm, where they came from in the original lab frame image, and also where they lie in this worm frame image. And so as an automated tool, we created this thing called the segmenter pipeline in collaboration with the Falks lab at UC Irvine, which takes in an input lab frame image, uses a UNet style convolutional neural network, which allows us to do sort of this image to image regression. So it takes in an image and it outputs an image. And then this creates three different outputs. We have We have our lab frame mask, which gives us the binary, like where is the worm, where is background. We get our AP coordinates, which tell us which side is the head and which side is the tail. And then these DV coordinates, which kind of you can think of as like the XY or the along the x-axis of our worm here. And then doing some output processing from these output coordinate maps, we're able to actually get these worm frame images. So the center line that I've shown you before is calculated using the DV coordinates image. So by finding the ridged line along the steepest gradient of these of the DV coordinates. The AP coordinates are used to orient the center line. So where is the head, where is the tail? And then the widths of the worm are found from the lab frame mask. I can't talk to you in a ton of detail about these just because of time, but I assure you it's cruel and it works pretty well. So the next step, of course, is to, now that we've found the worm in the image, we've been able to straighten the worms and account for positional variation. The next thing is to align the worms to a standard frame of reference. And this is because, like humans, not all worms are exactly the same. In fact, like humans, worms can come in a variety of shapes and sizes. So we need to be able to account for this sort of individual variation. And because these worms are different sizes, one can say, oh, why don't we just standardize the shape of a worm? You know, why don't we warp, stretch and squish these images of worms so that all of them are exactly the same shape? And what's really cool is that when we do that, we can actually create an average sort of bright field, what is a worm? So here is an example of averaging across 900 bright field images where the worms have been warped to a standard shape. And what's kind of cool is that you can start to see general anatomical features. So you can kind of see the pharynx here. Maybe you can see the vulva and you can kind of see the intestine, but it's really fuzzy. And this fuzziness indicates that shape alone is not enough to account for this individual variation. And this is because worms differ internally, but they have similar anatomical landmarks, which allow us to actually put all these worms into the same internal coordinate system. So here are 4 anatomical landmarks that every sort of standard adult C. elegans should have. So the 2 pharyngeal bulbs, the anterior posterior pharyngeal bulbs, the vulva, and the tail. What you can see here is that even though these images of these worms have been standardized, have been worked to a standard shape and size, internally, these anatomical landmarks do not line up. And this is what is causing that fuzziness. So the as a proof of concept, just to show you that aligning to these anatomical landmarks is really important and is really like the thing that is going to make it easier for us to analyze vast amounts of images of C. elegans at a time. Here, if we remember where we took those worms and we standardized the shape, if we take those same worms now and we identify the anatomical landmarks and then align it so that all those anatomical landmarks are aligned and then average those worms again, now we see very clear anatomical structure. So we can clearly see the 2 pharyngeal bulbs here. We can pretty clearly see the vulva, and the intestine is much, much clearer. And so this is just a proof of concept to show you that aligning to these anatomical landmarks is really important, and it's the way that we can analyze vast numbers of images. And of course, you know, as a computational biologist, the question that I'm always asking is, how can we automate this process? And to do that, I use that same sort of UNet style architecture to create this anatomical landmark identification pipeline. So how this works is that we take our straightened image from the previous step, we pass it through a dorsal ventral classifier to flip the image if needed, since worms crawl on their sides. And then we pass it through this UNet style architecture to create these output key point maps. These key point maps are basically an image representation of where the anatomical landmarks are along the length of the worm. And unfortunately, I don't have time to go into really like what this representation means and how we got there, because it was a long process to get there. But we settled on this type of image to image sort of analysis of, or regression of these worms in order to create these key point maps, because it seemed to be the best representation of the data that we like. And so using these key point maps, we can then find the anterior posterior coordinates of the anatomical landmarks, and then this will allow us to align all of the worms into that standard landmark aligned unit worm. So now all of the worms internally their coordinate systems are exactly the same, and you can treat every image of a worm exactly the same. So this is the pipeline that I've shown you and the different steps that we've created in order to get to the point where we can quickly analyze a bunch of worms. Now, what can we do with it? So it turns out that having these aligned worms provide many options for application. So once your worms are aligned to a sort of standard frame of reference, you can do so many things with them. And what's cool is that the system that we've created works on not only bright field images, which is where a lot of these identifications and classifications are happening, but you can apply them to a variety of different imaging modalities, provided that you have a bright field image taken in quick enough succession that the worm has not shifted that much between your two images. So with these aligned worms, you get for free anatomical variation because you are identifying these different key points, these different anatomical landmarks. So you can start to look at population level anatomical variation and also variation across time. So how do worms change over time? You can also look at different spatiotemporal expression patterns and fluorescent reporters. Again, if you take your bright field and your fluorescent image in quick enough succession such that the worm has not moved, now you can do a whole variety of different spatiotemporal things. And because we've aligned the worm to a specific coordinate system, now that we know where internally we know where every single point is on this worm, we can actually do tissue specific analyses with taking an atlas-based approach. So we apply the labels from our atlas to our aligned unit worm, and then this allows us to do sort of tissue specific analyses. And unfortunately, I don't have time to go into all the cool things that we've been able to do with this, but I do want to leave you with this tool set that we have developed. So in addition to the automated tools that I've shown you, we also have this sort of suite of tools that allow you to do your own curated sort of modular based approach. So here's a screenshot of the software that we've developed in the lab. It allows you to, I think it's great because there is so many things that you can do with it. So you can display your different images. If you have, let's say, that bright field and that fluorescent, you can put them here. You can tint the mask so you can actually see in your image here, where the fluorescence is. You can view the bright field image with the mask GFP overlay, so you can see what things are gotten out of it. You can also view your straightened worm and you can adjust the center lines. So let's say if the automated approach maybe fails a little bit, you can go in and you can manually curate and clean up anything that's happened. And in fact, I used this tool set when I was building all of my, when I was generating my ground truth data in order to make those automated pipelines. So the goal is that people who want to use this in the future and use the automated pipelines don't have to go through the pain that I went through to literally sit there and draw worms all day. What's also cool is that you can load many images so that you can just page through all of your different images. You can manually update the center lines and widths. You can indicate different landmarks or a variety of other things that you would like. And these are the different annotators that I used when I was designing these pipelines, but the lab has actually been able to use more annotators for, say, identifying death. So when does the worm die? We were looking at brood size. You can annotate brood size. You can annotate how big the worm is, how much it moves, a variety of different things. So these are the manual tools that help with sort of curating your data after you pumped it through the automated tool. And just something to leave you guys with, a little demo. This is the worm drawing sort of bit of it. So in real time, you can draw the center line of your worm and you can alter it. So I became very well acquainted with this and helped develop it when I was trying to draw a lot of worms to get those, when I was drawing lots of worms to build the ground truth data to actually build the automated pipeline. So I think it's really great. It's very modular. It's written in Python. and it's open source on GitHub. And so with that, I would like to thank the people who made this possible, especially Zach, my mentor, who let me just play around with different deep learning things, even though he is not necessarily the deep learning expert. And we got to learn a lot together. I also like to thank the Carrie Kornfeld Lab and Carrie especially, who was my sort of foster PI when the Pincus Lab was shutting down. I would like to thank my thesis committee, and then of course funding. And I would be remiss if I did not thank my coworkers. And I'm glad that they didn't make an appearance during this presentation, but they often do. So with that, I will take any questions.
Thank you, Nicolette. Do we have any questions?
I have a few questions. So
Okay.
I guess from your presentation, seems like your program kind of like analyze one worm at a time. So is there a way to kind of like analyzing multiple worms? And is there kind of a way to track, you know, a specific worm over time, or you have to manually kind of like tracking them?
So once you put it through the pipeline, you can actually, it runs really fast. So through the automated system, it runs really fast. Like once you have all of your worms, once you have all your worm images, you can just push it through the pipeline, and then it outputs like basically the like straightened out worm images that you want, and then you can do a variety of different analyses from there. In terms of over time, the worm corral system allows us to capture hundreds of individuals. So on a single slide, like I think you can get about like 200 individuals on that one microscope slide in our worm corral system. And then from there, we have an automated microscope that takes images of those worms every couple hours from hatch until death. So we're getting the time course data out of that. And then this pipeline that I've developed is kind of a more generalized pipeline just for like image analysis of worms in general. So once you get your data, you can just pump it through the pipeline. You get out the straightened worms, and you can do whatever you want with that.
Cool. Another question is about kind of like because basically you're kind of like compressing the three-dimensional information onto 2D. And I just wonder, like, have you ever kind of evaluated, you know, like how many, I don't know, like what's the best way to say it, just like how much information that you're like losing, you know, like when you're compressing, you know, from 3D to 2D. And because, you know, when you're taking 2D images, you know, like it's just, you know, like one picture at one time, but then the worms may move. And then if you take another picture, maybe like, I don't know, like a couple seconds later, like how much variations are you seeing for the same worm when you're taking like, you know, different images, maybe just like a couple seconds apart?
Right. So, I mean, that's obviously a difficult question to answer. That is one of the like, I guess, so because we are not taking like straight up videos of worms, we're taking like sort of these snapshots of the worm in a couple hours. There are systems that allow you to take videos of worms. It's just like to do that at the scale that we have. So there are people who do like videos of worms, but they're doing videos of like hundreds of worms, kind of like almost like a 96 well plate. So you don't get the same sort of microscopic resolution that we do. So, I mean, it's a trade-off, right? Like you can either take video or you can take snapshots. And so we chose to take snapshots, but with higher resolution, higher magnification images.